It's very likely that there'll be a multiple choice question or two about that, or at least to fill in the blank. So look at the different types of fossils. For whatever fossil we're talking about, whether it's a compression, an impression, one that has been replaced, complete tissue's been replaced by sediment or minerals, the right conditions have to be in place. We had the cow example in class where a flood could disrupt the bones. Something could come and eat the bones. We might only have a skull left because something ate all the other bones. If it is exposed area that doesn't get covered up, those will just degrade and won't have a fossil left. So to have specimens in the fossil record, things have to have the decency to die in a place they'll get preserved, and some things are just, they don't help us out. They don't die in places that help to preserve them. So the fossil record's incomplete. Well, yeah, that's because it takes very special conditions to form a fossil, and we just doesn't always happen, especially if an organism or a species didn't last long, wasn't very successful, or if there just never were very many of them. You know, if there were millions of T-Rexes, then we might find a few fossils. But if there were only a very small population of 1,000 or 2,000, well, our sampling size becomes much smaller, and the likelihood that species leave something behind becomes a lot smaller. So things have to die in a place to get them preserved. So let's talk about gene and vestigial characters. So vestigial and morphological characters. Morphological is the same as homologous. So you'll see that in different places. It's tomato, tomato. Same term, or different term, but it means the same thing. So a morphological or homologous structure is something like animals and their forelimbs. If I look at a bat or a cat or a frog or a whale, we all have the same bone structure in our forelimbs. If I take my arm and compare it to the bones in a whale's flipper, I've got an ulna, a radius, I've got a humerus bone, I've got carpals, metacarpals. All those bones are the same. They're just at a different scale. And with mine, I've got the differentiation of fingers, and the whale has the flipper, but the bones are all still there. That's homologous. The function is very similar as far as motion, being able to do things with that limb. When we start talking about vestigial characters, these things that may not functionally work the same way anymore, but we still see them. Some things to think about is like our tailbone. Why do we have a tailbone? Well, it's a vestigial character. We don't have a tail anymore. We can assume because of explanatory power. Remember, we just want to be able to explain things. Explanatory power says, well, somewhere back along the line, one of our common ancestors had probably a tail. It didn't do us much good, so we don't have one anymore. But we still have that base bone. Same for your appendix. We don't need that organ anymore, and so it's smaller and smaller and smaller until, you know, it doesn't form any, it doesn't provide any functional use. It can still make us sick, but you know, it's not useful anymore for what it does, but it's still there. That's vestigial. Another example we have here on this slide are the human head muscles. We actually have 3 muscle groups, main muscle groups in our head. Some of them we obviously still use, like our hyoglossus or our area epiglottic muscles. Your hyoglossus deals with tongue retraction, so you can sit there while you're listening to this lecture and stick your tongue out at the screen, and then put your tongue in your mouth, and your hyoglossus muscles just worked. Every time you swallow, you're using your area epiglottic muscle group. And all other mammals have these same muscle groups. We also have auricular muscles in our head. These are involved with ear wiggling. And if I was a rabbit or a deer or a dog, my auricular muscles are very well developed. I can sit there, I can turn my ears if I was a dog or a rabbit and hear predators coming up behind me, or listen for prey, and that's a very useful muscle group. We still have that muscle group, but we're kind of at the top of the food chain. And so we don't really need to hear predators coming up behind us all that often. And our prey, because of where our eyes sit on our head, we can track them pretty well too without needing to move my ears around a lot to do that. So I don't use those auricular muscles. Not many people can, some people can wiggle their ears a little bit, but not like a bunny rabbit. So that is a vestigial character. We still have the muscle group, we don't use it anymore. And the last thing that your book talks about, and that in your homework as well, is some of the gene clocks, the genetics that go on. And so the absolute best thing we can look for now, arguably better than things like just some vestigial characters or something like that, are genes. These are blueprints that all living things have. And the bases, the genes, the DNA bases that build up my gene blueprint, those letters are the same in every living thing. It's just the sequence of them are different. And the sequences undergo very small changes. A letter here, a letter there. I can track those. I can look at something that's very, very similar between all organisms. Let's look at the cytochrome C oxidase gene. It sounds horrible, but really all it does is help make energy. Well, if I'm a moth or a yeast or a duck or a person, I need to make energy. That's just part of being alive. It's something that all living things need to be able to do. They need to make energy. So I can look at that gene that every living thing should have and see how it differs from other organisms. So I compare my gene for cytochrome C oxidase to a pig. We only have 13 letters different in that story. If I look at me and a yeast cell, we both still have the same gene, but in that story there are 66 letters different. Well, from a particular clock standpoint, from a scientific standpoint, I can go, well, I must have a lot more in common with a pig because we don't have as many letters different than I have in common with a yeast. And then I can make that clade or cladogram diagram that's about in your books where, we use energy. So we have a common ancestor with the yeasts, but we've been separated a lot longer. And obviously I share a common ancestor with the pig as well. We haven't been separated on the tree as long as we've been separated from the yeasts. So genes are great, molecular clocks are great. It lets us look at the actual blueprint, not just a mouse, but the actual blueprint for what's going on with the living things. So that finished up for chapter 15, what we're going to talk about as far as evidence. So your test on Tuesday is going to cover chapters 1, 13, 14, and 15. That's 4 chapters. And a review will be on the D2L, if not Thursday evening, then Friday early afternoon sometime. So be looking at that review. Look over the questions from your homework again. Look over the quizzes on Eris. Go and look at those kinds of things and just practice. There will be 20 multiple choice questions on the exam. 5 matching. Those are with pictures. So look at images from your book. There will be 5 true and false questions to fill in the blank, so familiarize yourself with the terminology. And then you'll have 4 essays. We've had 4 chapters, and so it's a pretty safe bet to expect 1 essay question from each chapter. It'll be big picture kinds of things, things that help us understand the world around us, things you've been warned about in lecture. Hey, this is an important topic. So be sure to review those major points and stay safe, and I'll see you on Tuesday.